Good morning, everybody. Right. Oof. Feels like Friday already. Let's see. Get chat up. Okay, so today we have chi-squared test to do. I'm gonna get off my butt and feel like an old man today. Let's put the lights on. Fancy lights. Finally figured out how to use them. Bimapenko. Okay, so we have the chi-squared test to do. So hopefully you, actually, before I start, does anybody have any questions about p-values and the like, which is a super important concept to get? So now would be a good time to clear up any confusion or the like. For me to have some coffee too. Ooh, that's good stuff. Anybody have any questions? Questions? Nope. All right. So essentially what I'm going to do is I'm going to take you through a multi-step plan for success, right? So I always talk about these things. It's kind of a bit silly, really. But essentially, this is a kind of a list of steps that you need to follow in order to do a chi-squared test properly. And if you follow those, you'll get it right. So that's the cool thing about math, rather than just like general biology, is that, you know, as long as you follow the rules and you do what you're supposed to do, you can't get it wrong. At least the math I teach. So this is the way that I found works pretty well with students. All right, hang on a second. Aha, more pens. All right, start again. Forgot that that one had gone out. There you go, that's better. All right, so the first step, and this is really the most important step of all, right? Because it's all based on this, is what is your hypothesis, right? So the very first thing you have to do is frame your hypothesis. Right, and really, to be honest, you want to do that before you even start the experiment, right? Because that's what will dictate the design of the experiment and the questions you're asking and how you do it. So really, this should already be done, right? But in many ways, we do this, at least in this context of doing these chi-squared tests, a little bit after the fact. But in lab for the Fly linkage project, you'll be doing exactly this, right? So you'll be following all of these steps to do the analysis for your linkage results, right? So you're going to actually be doing this kind of with real data, right? Data that you collect. So the reason why you frame your hypothesis is that gives you your predictions or expectations. Well, and really that gives you your expected values. And these are very important. They are central to how a chi-squared works. So you can see that if you don't have your hypothesis framed correctly, you don't get the correct expected values. And this is actually a really common thing that I find in the Fly linkage lab reports that I ding people for because it's wrong, is that they will give a particular hypothesis. These genes are linked. And then they will generate expected values for genes not being linked, right? And so that gives completely the wrong result in terms of chi-square value and p-value. And so it just makes a mess, right? So you have to be very clear, what is it that you're testing? And if your hypothesis is supported, that one, hard to do it in reverse, that will give you certain expected values. So you have to be consistent, right? For the Corn Genetics Lab, I'm not sure what the adjuncts are saying, but what I always say is that, you know, I'm not going to be doing the chi-squared to see if your numbers are right. Really, what you're going to be marked on is, are you being consistent? Right? Are these the predicted the expected values that you would expect for this hypothesis, for this particular cross? Is this the chi-squared value? And if you get that chi-squared value, then you should get a certain p-value, right? It's in the table. So as long as you're consistent throughout, even if you get a really weird result, you get it right, because that's what we are caring about. But in real life, you got to figure this stuff out first. Okay, super important, really. Okay, so the next thing you do, step 2, is calculate your expected values. Right. Is this all coming through like the right way around? Because it isn't mirrored for me. Is it right way around for you, or are you trying to read this all backwards? Oh, glad that's working. I should have asked that before, shouldn't I? Anyway, all right. So again, from teaching this a bunch, I know that one of the mistakes that students make is they don't use the correct number to calculate their expected values. The number you should use is the total of your observed or of your observations, right? Right, so that's really important. So if you're doing a monohybrid cross, right, and you're testing to see if the F2 match what you expect, then it's the total for both the dominant and the recessive phenotype added together. If you're doing a dihybrid cross, then it's the total for all 4 phenotype combinations. Whatever it is that you're doing, however many different things that you're observing, you add all of them together and use that number, right? Because essentially, if you expect a half of your observed to be X and you score 100, then it's going to be 50 because it's half of 100, right? So you've got to work off of the total observed. And then you use your predictions to calculate what you expect. So let's put it here. Right, so if we've got a monohybrid cross, the F2, what fraction of the offspring or of your observed would you expect to be dominant in the F2 generation of a monohybrid cross? What fraction should be dominant? 3/4. Fabulous, Isaiah. That's exactly right. So if we're looking at the dominant, it's going to be total observed times 3/4. Right. If we're looking at the recessive, it's going to be the same times what? What fraction of the total in the F2 would you expect to be recessive for a monohybrid cross? Yeah, it's 1/4. Exactly. Right. Good job, everybody. right? And so you can see why it's so important to figure out what it is you're testing, like what's your hypothesis, because those fractions or predictions give you your expected values. And if your hypothesis changes, so do your expected values. So here's another way of thinking about it. And I try and get you to think about this in as many different ways as possible when I have the time, because that's how I'll be testing you. So for example, if you were doing a test cross, single gene test cross, and your hypothesis was that the unknown dominant is heterozygous, what would your expected fractions be for the dominant and recessive phenotypes? One gene test cross, hypothesis that the parent, the unknown, is heterozygous. Pardon me. Got to get you thinking, right? Get you out of your comfort zone. What ratio do you expect? So
Can you repeat the question, sir?
actually, let me type it down. See if I can do this one-handed. Oh, one-handed. Hypothesis is that unknown dominant, bloody hell, it's hard to type, is, I'm not going to type heterozygous, that's just too miserable with one hand. Question mark. Yeah, so that's up for everybody. We have a think about that. I'm going to have some more coffee. Sweet nectar of life. Manna from heaven. Anybody get that film reference? It's a bit of an obscure one of my favorites. Come on, someone's got to watch that film. It's a classic. Let's see if I can do a proper Aussie accent. Sweet nectar of life, manna from heaven. How about that? Anybody? Yeah. It's Finding Nemo. It's the crabs in the sewer pipe, Sydney Harbour. Anyway. Okay. Nope. That's a monohybrid cross, Stephanie. Good try though. We're not doing a monohybrid cross, doing a test cross. Yeah, awesome. Cool. So if you were doing a test cross, you'd be doing the experiment differently, right? Because your hypothesis is different. So the way you test that hypothesis is different. And so instead of having 3/4 and 1/4, it'd be 1/2 and 1/2, right? So that's why it's so important to get this right, because if you get that right, then these will be right. If you get that wrong, these will be wrong, and you'll come to the wrong conclusion because your chi-square test will tell you something that's wrong. Does that make sense? Cool. Can't believe no one got that Finding Nemo reference. I'm super disappointed. Okay. Oh well, can't get them all right. It's not Pokemon. So the thing now, yeah, it's one of my favorite films, I have to say. I've got lots of favorite films, but that's one of them. So yeah, there's lots of other quotes I can give you from that. Right, distracted, focus. So now we do our actual chi-squared test, right? So for example, let's give some numbers here. F2, let's say 100 individuals scored. Always use round numbers because they're easier to do in your head. Okay, so in this case, our expected values would be 100 times 0.75, 100 times 0.25. I'm a bit like allergic to fractions, I have to say, for decimals. That is 75, that is 25. Right, so those are the numbers that we're going to be using for the expected values. Right. So now we need to calculate our chi-square value. So use like a funky X, the 2 on the top. This is that, which means sum of observed minus expected. So these are what we actually record to get our total of 100 minus what we think we should get, which is calculated using that number and our fractions. Now, obviously, doesn't matter how many you have, but you're almost certainly going to get numbers which are higher or lower than you're expected. So you're going to end up getting negative numbers. So once you do that, you square it. That'll turn everything positive. Now, because this difference, so this is essentially what you're doing is you're trying to figure out what's the difference between what I think I should get and what do I actually get. Yeah, it's kind of like what I think I should get in an exam and what I actually get in an exam. Hopefully those two differences are very, very small. Oh, actually probably shouldn't go down there as an analogy. But anyway, so thing is though, this difference will be bigger the bigger the numbers you're looking at. So if you're just looking at 100, you might have a difference of say 2 squared. If you're looking at 10,000, you might have a difference of 200 squared, right? Much bigger number. And so we need to account for the absolute sizes, right? So that we can compare, small sample sizes with big sample sizes. And so we divide all of that by the expected, right? And so that will allow us to basically scale this difference and make it comparable for, n is 10 versus n is 2 million, right? You'll still get the same values for the same relative difference. Does that make sense? Cool. It's all about figuring out how big the difference is between what you expect and what you actually get. So because this is sum of, essentially what that's telling you is you do one of these for everything that you score. Every different condition, phenotype, variable, whatever, right, that's in your experiment. So in a monohybrid cross, we've got 2. So you're going to be doing 2 of these calculations. And essentially, that would be observed for the dominant minus expected for the dominant squared over expect for the dominant plus observed for the recessive. Oops. Don't copy that. I don't know what's going on in my head. Expected squared over expected. All right, sorry for the shaky cam. My arm's getting tired. It's like watching one of those cam recordings of cinema releases. So let's make up some numbers. Where can we put this? That'll do there. So what you actually observed was 77 dominant Probably better use just 1. Not sure that makes any difference. And 23 recessive. Right, so those two add up to your 100, your total observed. We're going to fit those into this equation. Right, I'm going to do this one for you. Then I'm going to give you another one to do as a test, and then we'll do a different one. So we're going to run through this one first, and then we'll do a couple of other practices. Okay, so dominant, that's our observed for the dominant. This is our expected for the dominant. Squared over the expected for the dominant is 75. Plus, because it's a sum of, where we're at, 23 for the expected for the recessive. Ah, man, come on, Matt. My mind on animated films for some reason. Oh, quick, I was talking about this in lab yesterday. Has anybody seen Love, Death and Robots on Netflix? I'm a big animation fan, so it is super awesome. If you haven't seen it, go check it out. Some of them are, yeah, they are. Oh, I've watched some of them like 5 or 6 times. They are fabulous. One of my favorites is Lucky 13, but there's also a really cool one about what happened if Adolf Hitler had been killed at certain stages through his life. It's really, really funny. Really bizarre and totally not appropriate for kids as well, so don't watch it with them. Anyway, so 23 minus our expected, 25 over 25, right? I'm going to need a little bit of help with the calculator in a second. So that one's easy. It's going to be 2 squared over 75 plus 2 squared over 25 equals 4 over 75 plus 4 over 25 equals what? Oh, sorry, pointing at the wrong thing there. Could someone work out for me what this over this is and what this over this is? Is that the total, Bianca, or just one of them? Okay, so that's 0.53 plus 0.16. Is it that way around, or is it that way around? Doesn't actually matter in the end. Yeah, okay. So together that is 0.69. Right. And that is our chi-squared value after all that. Right. Other people check Bianca's work. No, I don't trust you, Bianca, but it's just good to double check. Doesn't hurt to make sure you put everything in the right place. Ah, okay. That's okay. No worries. That's why I asked Bianca, because this should be a much smaller number than this one. Oh, dang it. This should be a much smaller number than this one. Yeah, those zeros are really important if they're in a particular place. It's not going to make a big difference in terms of what we're working on. Yeah, have coffee before class, right? You don't want to wake up after we've finished. So that is what, 2.13, I think. Yeah. Oh, just saw that, Stephanie. Cool. Okay. It's going to take a picture of this and then. Oh, yeah. All right, so I've left that up there because we're going to be going back to that bit there. Where did my pen go? There we go. Okay, so we've got a chi-squared value, but this on its own isn't enough, right? The reason being is that the more of these calculations you do, the bigger this number is going to be, even if it has the same difference, right? If that makes sense. So if you do 2, right, you got small value plus small value, right, equals small value. If you do 200 of these, which would be miserable, but let's say you did, for whatever reason, if you had 200 of these, then this value, well, there we are, is going to be larger because you have 200 of those to add together, right? So just as we had to kind of scale this difference by dividing by expected, we have to scale this value depending on how many different conditions we have, right? To figure out whether we have a significant difference from what we expect or just a random, doesn't matter difference. Okay, so go and start back at the top. Fourth step is calculate number of degrees of freedom. Freedom, DF, right? My wrist is actually starting to hurt. At least start doing like some wrist curls or something before class, getting training. Thankfully, I have a fairly light laptop. This would be miserable if it was one of those gaming laptops. And so that is n minus 1, where this n, it's kind of the same as n as in sample size, but instead of number of things that you observe, it's the number of different conditions that you observe. Conditions, phenotypes, etc. Right? Or, another way of doing it is the number of calculations you carry out. So here we're carrying out 2 calculations because we have 2 phenotypes. So our degrees of freedom is 2 minus 1 equals 1. Right? So this is the second value that we need, right? We need 2 values, chi-squared value and the degrees of freedom. With those 2 values, we can calculate the significance of that difference. Right. And to do that, we use chi-squared value, degrees of freedom, and chi-square table or chart. Right. And I'm just going to sit down, rest my wrist, and show you that chart. Oh, ooh, that's better. My aching bones. All right, so and this should be in your exams and quizzes folder in the lecture part of Blackboard. Come on. Pardon me. I'm just going to share my screen. Okay, so here it is. I'm going to leave the chat up over there. So you can use a table, you could use a chart, right? And it's up to you. So I actually work off the table better. If you're more mathematically inclined, the chart is perfectly usable. I think it's just easier to teach it using the table. And so the table has three different values in it. It has your degrees of freedom. It has different chi-squared values, which is what are in the columns and rows, for that any given p-value and degree of freedom. And along the top are the probability values, the p-values, right? So one thing to remember again is the larger the p-value, the less significant the difference. Because the p-value is the probability that that difference occurred by chance, right? So oh, come on, fingers. And so the number we care about as scientists is this one here, the p-value of 0.05. So a p-value greater than, 0.05 is non-significant. P-value of less than 0.05, decimal place, equals significant. I don't know who that is. Hopefully they'll leave a message. So this is the column that we care about. And so you can see that as the chi-square value gets bigger, is the difference between what you expect and what you observe gets bigger, then the significance increases. The p-value gets smaller, right? Because the probability that you'll observe that difference by chance gets less and less, the bigger that difference is, right? And so the line in the sand is this column here, p of 0.05. So if you're, this is worth typing out too, if your chi-square value is greater than the one in the equals 0.05 column for your DF, then that tells you that you got that difference by something other than chance. And essentially, that means that the underlying hypothesis of what you should expect was wrong. There's something that you're not accounting for, right? That's not just random biological noise. It's something meaningful. Like your assumptions were wrong, or you did the cross wrong, or who knows what, right? There's something that's happening which is affecting your results. Now, if it's lower than that value, that chi-square value in the 0.05 column, then that tells you that your result is not significant and occurred by chance. And essentially that means your hypothesis was supported. Any questions about that before we move on? It's kind of a lot to take in, and it's weird because the bigger the chi-square value, the more you care about it, the smaller the p-value, the more you care about it, right? So it's not necessarily intuitive. Any questions? Okay. So let's go back to the chi-square value that we got. Okay, well we'll do a practice in a second, Stephanie, and then you can figure out what makes sense and what doesn't. So we have two values. We have chi-square equals 0.213. That's right. Degrees of freedom equals 1. And so we're looking at this top row because of our degrees of freedom 1. And we're looking to see where our chi-square value fits. And so you can see that the chi-square value is less than the one in the very first column. So it's smaller even than the one in the 0.5 column. So p-value is greater than 0.5. In other words, the probability of getting that result by chance is 1 in 2, 50%. So really high. Basically, it means that if you do this experiment again, there's 50% chance you'll get a different result. You know, so there's nothing that's actually forcing that difference that you observe. It's simply because random noise or sampling or sample size issue or whatever. So to present your, let's get this smaller. Here we go. To present this, you have to write it out in a very formal way. You have to write your chi-square value, degrees of freedom, and then your p-value. That is your chi-square test. I'm going to take a picture of that. Now I'm going to give you an example. Just going to take a picture first. Okay, so let's do one which should maybe illustrate things differently. So I'm going to give you your data. Let's see. I'm going to tell you what we did. Monohybrid cross. Observe F2. See, what would it be? Let's say 110 dominant and 8 recessive. Calculate chi-square, df, and p-value. This is a kind of question you will get in the exam. Although it may be for a dihybrid cross, right? So it's a little more complicated, but same kind of idea. Where are we pointing? There we go. Okay, I'm going to give you a couple of minutes to get started on that, and then we'll go through it before the end of class. I'll just actually type that in the chat as well, so we have a written record of it too. Written? So I'm from Cockney. Okay. Okay. Give you until, well, just a couple of minutes. Sorry, kind of spent a little longer on something than I intended. Some parts of this. Okay. Sorry, it's a little bit of a whistle stop tour, but you can go back through this as well yourselves. Okay, so first off, using our plan for success. Right, I've already given you the hypothesis, right? We have a monohybrid cross, so we know what to expect in the F2s. What are your expected values? What do you need to calculate your expected values? Come on, peeps. You're not going to have any help from me during the exam, unfortunately. So give it a stab. We've got 4 minutes left. Okay, so I'll give you a hint. It's a good stab. I think that you might have something not quite right going on there, as I'm not sure what. So let's start from kind of like wind things back a little bit. What's your total observed? Right, it's 118. And of those 118, what fraction would you expect to be dominant? Right, so it's 3/4 of that total. Right, so your expected dominant is 88.5. Expected recessive, oops, equals 118 times a quarter, right? Because that's a fraction that you expect to be a quarter, equals, what do you put? 29.5. Great. So now we need to calculate the expected, well, now we need to calculate the chi-square value. So for the dominant, it's going to be our observed, which is 110, minus our expected. Yeah, that's right, Bianca. 88.5 squared divided by our expected, 88.5. So what's that value come to? Let's see if I can do it on my clunky on-screen calculator quicker than you lot can. So 110 minus 88.5 is 21.5. Yep, let's see. See if I get the same one. Squared is 462.25 divided by 88.5 equals 5.22. Yeah, awesome. Right, so you all got the same result. That's great. So the recessive, there's only one C in recessive. I don't know why I typed it 2. Is, and we'll finish this, right? Even though we're going to go a minute or two over, right? It's going to be what? 8 minus 29.5. Apologize for enormous fingers. Squared divided by 29.5 equals what? 15.67. So I'm going to take your word for that because time's running out. So the chi-square value is 5.22 plus 15.67 equals what? That should be like 20.87. Yeah, 20.89. 20.89. Great. Degrees of freedom equals what? 1. So now we go to, actually, let's get this up first. You don't want to read about that. Okay, so let me bring up the chat again because that has all the stuff in it. Okay, so same row, 20 degrees of freedom of 1. Now we look on here and say, is our chi-square value 20.89 larger or smaller than this value? Right? And the answer is yes. The value is actually higher than the largest value we have on this chart for a degree of freedom of 1. So our p-value is less than, well, let's top it out properly, 0.001. So the difference observed is highly significant. Therefore, our hypothesis or assumptions were wrong. And you would type this out as chi-square equals 20.89 d, oops, not FD, d f, come on, equals 1, p-value 0.001. And so essentially here you got way fewer of the recessive than you would expect. So something was happening to result in a lower survival or development or whatever of the recessive phenotype, right? So things weren't following Mendelian assumptions and rules, even though you did the cross correctly. Okay, so I would strongly suggest you practice this with both monohybrid and dihybrid crosses, maybe even test crosses too, right? You just make up numbers and work through those different steps, and you'll be able to bang one of these out in a couple of minutes or less. Okay, cool. I've gone way over time. Apologies about that. I'm going to sign off quickly and let you get on to wherever else you need to go. And I'll see some of you for lab. So practice, practice. You'll have questions on this on the exam, guaranteed. Cool. All right. See you all next time.